Hello, welcome to another video from the conscientious biologist Ben Gallagher. This is the second one in the protein series, and as I said in the first one, I think understanding of proteins, what they do, how they're formed and how important they are to a cell's functioning is the most important concept to get your head around in biology. So this is a really important one. It's aimed as always at GCSE level, and please do subscribe to my channel if you enjoy these videos. So let's start with the most basic question of what are enzymes? And we're going to run through the key points of that. Now, there's a reason why I've put Pac-Man up above my head, and that'll become clearer as we move through the video. But let's get the key points up first. Well, the most obvious thing, it's in the proteins unit. They are proteins. It's always really annoyed me when I pick up a class in year 10 to take them through their GCSEs, and we'll come to enzymes, and I'll say that enzymes are proteins, and they'll look really shocked. And they've studied it very much as a separate little thing, that enzymes are this totally unique thing in their own right. Well, remember, enzymes are just proteins, and proteins do everything. Yes, enzymes are incredibly important proteins because they carry out the reactions, but they are still just proteins. They're biological catalysts. Now, the word catalyst you might be familiar with from chemistry. A catalyst is something that makes a reaction happen far more easily. From a chemistry point of view, it lowers the activation energy, so the reactants can more easily be turned into products. But for biology, that's just the key term you need to remember. They're biological catalysts, they're biologically produced within cells, and they make reactions happen. They're the main feature that drives metabolism. And if you remember back to the cell biology lessons, metabolism is kind of the defining characteristic of life. It's all the reactions going on within a cell. Because enzymes are the main feature that drives metabolism, what we're saying is they do most of the building and breaking within a cell. We'll come on to some details about that a bit later. Now, the most common ones you will study will be digestive enzymes, which is why so many students that I pick up, when I start talking about enzymes, they go, oh yeah, just break down food. They do so much more than that, okay? Almost all biological reactions use enzymes. They're incredibly important. Now, I've just put up this picture here. I showed you the Pac-Man before, but this, look, is a Lego version of Pac-Man. Now, remember that from the previous presentation you should have watched. We said that proteins are built of their building blocks, and we linked it to Lego, how you can break one Lego thing down, reuse the building blocks to create something. So I've just put that picture up there of a Lego Pac-Man just to try and help you remember back to last session. But of course, when we're talking about enzymes and proteins, they're not really made out of Lego. But if we wanted to represent an enzyme diagrammatically, we could just put something like this. Look, it's kind of the same as the Pac-Man version. I've just made it more simple and drawn it sideways and just flat. But you'll notice about it, it's still got that mouth area. Now, that mouth area on an enzyme is called the active site, and it's the active site where the building and breaking molecules occurs. So this is the equivalent of when you're building something from Lego, you use your hands, you grab bits, you pull them apart. Well, enzymes don't have hands when they're doing their building and breaking to do their, to catalyze their reactions. They have to use that mouth area. So it's as if I gave you a challenge of having a load of Lego and building me a Lego house, but only using your mouth, okay? It'd be really hard for you, it's easy for an enzyme. So to consolidate your learning from proteins 1, we're going to apply the protein synthesis theory to enzymes. Because remember, they are just proteins. So enzyme formation is still just protein synthesis. So really quickly to recap that. Remember, to build proteins, you need a ribosome, and the ribosomes need to follow the instructions in the DNA to put together a whole long chain of amino acids, and that chain is called a polypeptide. Now, of course, that long strand can't really do anything yet. What it needs to do is form a shape, because shape determines function. So if I just shrink it down there, but what you need to remember is proteins are way longer than just that string of emojis that I've used. So if you imagine it's etc, loads of those in a line. And just to make it simpler, I'm going to replace that with just one straight blue line. So please understand, if you haven't seen the proteins 1, this won't make as much sense, but please just understand that long blue line is just representing the long string of amino acids, the polypeptide. Now we said to have a function, it needs to achieve a shape. So it folds in a very specific way due to the specific sequence of amino acids. And that sequence, of course, was dictated by the DNA. So the ribosome looked at the DNA, put the amino acids in that specific sequence, which made them fold in a very specific way. Now look at how I folded it over there. It's just kind of a tangled mess. But that tangled mess is now an enzyme. It can have a function. Now we never draw them as tangled messes like that. We just kind of draw the outside of it, the structure. But you can see if I just draw over the top of it there, it's retained the shape of that folded polypeptide. We just made it look like a bit more of a solid lump. But there we go, that's it. That's my enzyme. And link back to the previous thing with the Pac-Man, you can see it's got its mouth. It's got that active site, which, as we said, is where the building and breaking of molecules occurs. So let's look at enzyme action now. How does the enzyme do those reactions of metabolism? So this is relatively straightforward. If I've got my enzyme there, you can see the active site. Here's the thing that it's got to change, either break it down or build it up. In this case, we're breaking it down. Now, the thing that an enzyme reacts with is called a substrate. Now, if you look at the two up there, you can see that the shape of the active site kind of matches the shape of the substrate. They're not identical. It would be crazy to say they're identical. They're quite clearly not. They're almost the opposite of each other, so they can fit together perfectly. Just like a key and a lock, they will fit together. So because they match, they can form this. Okay, this is called an enzyme substrate complex, where the substrate locks into the active site, so the enzyme's kind of now bit down on it, and then the enzyme can use its active site to break it down into the next pieces. And those next pieces are called products. That should be fairly obvious. You produce something, they're called products, or manufacturing makes products. So we've got a basic enzyme reaction there. The substrate fits in the active site, forms an enzyme substrate complex, which breaks those down into products. However, it doesn't just have to happen in that way. Remember, we said that enzymes can build stuff and break stuff. So that top one, that was breaking stuff. But if I repeat the diagram here, but flip the arrows around, you can now see I've got two things going in, these things right next to me, and they're going to turn into that one. So these two are now the substrates. That's the single product over there. So depending on which way the enzyme goes, will change what kind of enzyme it is. These ones at the top over there, those are catabolic enzymes. Catabolic meaning breaking stuff down. This bottom diagram is anabolic. Okay, anabolic means making things. So some enzymes are catabolic, they break stuff down. Some are anabolic, they can build stuff. But when you look at catabolism, which are the catabolic reactions, and anabolism, which is the anabolic reactions, when you add those two together, so all the breaking stuff, all the making stuff, we've already said in previous videos, that's metabolism. And metabolism is the key to life. Just as a quick little aside point that might help you to remember some of this, you may have heard in athletics and sports about anabolic steroids. Well, hopefully that term might make a bit of sense to you now. An anabolic steroid helps you build up and make more muscle, which gives athletes an advantage, which is why anabolic steroids are banned. Nothing to do with enzymes, it's just a useful way to remember that word if anabolic steroids is something you've heard of in the news. So now that you know that basic enzyme mechanism, we can just introduce this one last term. This is substrate specific. It's a really important term when we're talking about enzymes. So looking at the enzyme that I've put over there, it's the same one that we've used previously. You can see that they match each other. That means when the substrate moves to the active site, they can lock together because they fit perfectly and form that enzyme substrate complex. Okay, the shape of the active site and the shape of the substrate are complementary, and that's the really important key word. If you look at the lock and the key that I've put over there, a lock and a key are not the same shape. If I took a padlock and a key and showed them to my 3-year-old and said, look, same, he'd look at me like I'd gone mental. There's no way that they're the same. However, the internal mechanism of the lock matches the key. They're kind of mirrors of each other. The word for that is complementary, and that's a really important key term. Okay, so that enzyme will fit that substrate because they have complementary shapes. But if I put a couple of different substrates up there, so a sort of rectangular substrate and a triangular substrate, quite clearly those two substrates aren't going to fit in the active site of our original enzyme. It doesn't mean they can't be broken down, but it means they must have different enzymes to do that. So that substrate could have an enzyme that's like this, which means it can fit into the active site of that enzyme, and that means that one can break it down. Now, please remember what we said about proteins and protein synthesis. That new enzyme that can break down that rectangular substrate is only that shape, and its active site is only that shape, because of how the polypeptide, the long amino acid strand, because of how that folded. And it folded because of the sequence of amino acids, and that sequence was set out by the DNA. So the DNA gives the sequence of amino acids, which controls how it folds, which gives it its shape, which determines its function. Those are really important points, okay? If we want to break down that triangular substrate, we need a polypeptide that's folded in a different way. So it must have had a different original sequence to fold in that way. So we get a triangular shaped active site to fit a triangular, or to be complementary to a triangular shaped substrate. So every substrate can have its own enzyme. Every enzyme is specific to one substrate. So we can use that term in a sentence, and it's that enzymes are substrate specific. That's why the title is substrate specific, because enzymes are substrate specific. Now this is really important, and we can pull this back to the cell biology lessons. What that means is that a cell can be very specific in which enzymes it creates. It doesn't have to create all the enzymes that are encoded in its DNA. And that allows it to be very specific in which metabolic reactions it performs and what it does. If you've got a cell that only wants to do a certain number of metabolic reactions and do a certain few things, it only makes the enzymes for those things. It doesn't make the enzymes that do everything else. And that means that by controlling the enzymes and all the proteins it makes, a cell can differentiate and be specialized. So this links back to one of the cell biology lessons on differentiation in specialist cells. It's all about which proteins, which enzymes a cell makes that mean it can have a specialism and become differentiated. Okay, so what we're going to do now is we're going to summarize this, but we're going to do this in a really, really useful way. It really upsets me every single year when I have great students who know all the knowledge, it's all in their head, they've understood all the concepts, but then it comes to the exam and they struggle to score marks because they struggle to demonstrate their knowledge. So I always do a lot of work with students on that. And let's have an example of that now. If I put a question up of what are enzymes, it's a very broad question. But if I said to you, I want you to write me an answer that would be worth 15 marks, 1-5, you might go into panic mode and think that's a huge amount of marks. I've got to write a whole essay. What a nightmare. But you don't. And this is what I need you to start to understand. You just need to write brief, concise, but detailed answers. And that's what I'm going to give you an example of now. Now, as I put up each point, you'll see over to the far side of it in orange, the number of marks that line would be worth. And you'll see that the number of marks correlate to the number of details that I include. So starting off, enzymes are proteins which act as biological catalysts. 2 key words there in capital print. To convert substrates into products at a rapid rate, 2 more marks. Enzymes have very specifically shaped active site. Definitely a mark for active site. A lot of examiners and exam boards might give you a mark for shape as well, because it's the shape of all proteins that's so important to their function. But enzymes have a very specifically shaped active site, created by specific folding of the polypeptide chain of amino acids, 3 marks, which is complementary to only one substrate. This makes enzymes substrate specific. Enzymes can be catabolic, so break things down, or anabolic, build things up. The total of both these reactions give the cell its metabolism. And by controlling which enzymes and proteins it makes, a cell can differentiate and specialize. 2 more marks for those. That's your 15, for the total over there in the bottom. Now that's really not much writing. You look at everything that's on this page there. That's not a lot. In fact, if I set you the challenge right now of grabbing a pen and paper and just copying out what's on the board, you'd probably find if you were using standard A4 paper, be a paragraph about that big. It's very, very little writing for 15 marks. And this is what I really need you to start practicing and getting your head around. Please start thinking about answers in terms of clear, concise little bullet points that contain huge amounts of detail. That's what shows your knowledge. That's what will get the marks. Okay, that covers everything you need to know really about enzymes, just from a structural and functional point of view. But you need to extend your knowledge now by looking at factors that affect enzymes. This is the next video, which is proteins 3. This is where you get loads and loads of exam questions on this, because you have to apply the knowledge from this video to answer the questions that come up in the next one. So head there next, please. As always, please do subscribe to my channel on YouTube if you haven't done so already. Like this video if it's been useful. And I do now have the Facebook page. Head there, there's a lot more on there than just GCSE biology stuff. Thank you.